So this is about the trials that I've been running at Ataxia and what kind of placebo effects we've seen in there and what we could learn from this. I want to have at least one slide on the mFARS score because these trials, they use different versions of the mFARS, and in order just to avoid the confusion, I'm going to give a brief comment on that. Then how did I estimate the placebo effects in these studies? Then the main part, I will talk about the studies. At the end, look where I have the data at the subscores and how they reacted with regards to placebo effect, and then summarize. So on the mFARS score, just we're on the same page. The mFARS was initially a concept that covered 4 domains. It had a staging, an ADL, the neuro exam, and time measures. And for those who don't know or haven't noticed, all the other 3 are used in the spinocerebellar ataxia, just not the neuro exam so far. And I think that should be changed. Second, the neuro exam is sometimes called the mFARS 3. So that's just because it was the 3rd domain. It doesn't mean anything else, just to avoid the confusion. The neuro exam was then reduced to what we use now, the modified mFARS or mFARS. That followed an FDA interaction, so that's not known in detail, but the FDA clearly favored this reduction, and later it was shown that this improved the properties of the scale. So that's what we currently do. The most important component of the mFARS is the mFARS E. It's the last subscore. It's also called upright stability. So these 3 you should keep in mind. The whole neuro exam, the mFARS, and the upright stability. These are the 3 most important. Then there is a slight catch in some of the historical studies. Some of the items were omitted. There's a whole story around this, but I spare you those details. Some items that covered stands with eyes closed were not used in several studies. So that's why those have different versions of the mFARS score. Every time there is a number behind it, remember that's the reduced score. It's always 8 points less. So the 93 becomes 85, 36 becomes 28, and so on. I think with regards to the placebo effect, that doesn't really matter because we're looking at change from baseline, and the amount of placebo effect is not really impacted. So then I have a couple of slides to explain that in more detail. I think we can skip those. Whoops. And I come to how I try to estimate the placebo effect. Funny enough, I have the same slide as Annette, just my paper is 8 years older. People do recycle their stuff. What I did is I looked at natural history studies as comparators. I have these data. Then I took the placebo response from the randomized controlled trial and compared these, and that's the placebo effect as it's shown here on this slide. But please note, this is a rough comparison, and you will see that in my summary later on, and I see everyone has the same things on their minds. We know that there's a lot of things that can go wrong, but at least to roughly compare, I thought, let's just do the exercise once. I'm using these 8 studies here that have been conducted between 2005 and 17. It's arguably the most important ones that have been conducted in FA until then, of course, and afterwards, MOXIE and MOVE-FA, I would also comment on, but not within this analysis. When we do this, we need to keep in mind that the decline in FA, in mFARS, is age dependent. So the population you pick based on an age, and that age of the population is the most important determinant of the decline. And you can see that in this graph. These are age brackets, and you can see with higher age, the decline is decreasing. So I just looked at what age group did this study recruit in, and what kind of change does that produce in the natural history study? It's a very simple thing. And I'm going to start with the first idebenone study. Internally, we call this Nicosia. It was run at the NIH, an investigator-initiated study. I can see that in all outcomes, whatever we look at, you have very nice dose response curves. On the left side is always the placebo group, and then you have low, intermediate, high dose. And I think this looks very convincing, and that's why everyone got so excited about idebenone. In the total group, It's a little bit small here, but the placebo response was a bit more than 2 points. No, I should say the improvement in the placebo group was a bit more than 2 points. And depending on what natural history study you look at, this corresponds to 1.4 or 1.8 points of decline in half a year. This is a very severely affected population. And then you add this up and you end up with more than 3 points of placebo response in half a year, which is huge. Then in there, what they call ambulatory population, that's a certain restriction on the ICARS, which was used at the primary endpoint in this study. You see that ambulatory patients don't have this placebo response or improvement again. You see, I'm already making mistakes with the language. But if you compare these 0 points, you end up with a placebo effect of 1.6 to 2.0. The response is about minus 0.2. So that's what I added up here. Again, already by looking at subgroups, the placebo effect is hugely different. Now, this, of course, got everyone very excited and is basically the reason why Santhera started their idebenone project, or was a very good reason for that. And they looked at exactly this subgroup in a study called Ionia. And indeed, here, the placebo group declined after 6 months. And still not reflecting the full decline of the natural history study, which is, again, depending on which you look at, between 1.5 and 2.3 points. So the placebo effect here is much smaller, 0.7 to 1.5. And yeah, I think that's already when I thought, wow, that's a big difference. Okay. The third study with idebenone was Mykonos, and that was a huge study, like 240 people, 70 per group. And the completers were a little bit less here, around 50 or 55 per group. And this study recruited 8 years and older, everyone 8 years and older, no functional restriction. And that makes the study very easy to recruit, but also very, very difficult to compare with anything. And you can see the placebo group, I took this from clinicaltrials.gov, it's not published yet, declined by about 0.9. Now, if I take everyone in the natural history study, the average decline is about 1.8, but I don't think we can compare these. And unfortunately, there's little information about these patients available in the public domain, so we can't really subgroup them or look anymore closely. It would maybe be worth the effort if we get more data of it. Fourth study. This is the deferiprone study. It's an iron chelator that was enrolling a very broad and diverse population between 7 and 35 years. I don't think anyone would do that again today. An FA patient with 7 years is not comparable to an FA patient with 35 years. And in addition, they had a very small placebo group of 11 people. And in the results table, they split this even up into early patients, ICAS less than 31, or late patients, ICAS higher than 31. And you can see the placebo response here in the early patients. And again, I say placebo response. The change from baseline in placebo was 0.6 points. And in late patients, it was minus 0.4 points. So they improve. This is what we often see in milder patients that are older. We see what we call a regression to the mean. They just improve a lot after half a year. So I don't know if I should put a number on this based on 5 or 6 patients, but if you do, you end up again with these 2 points after 6 months. Then there is a study that was done with the epi compound. Today it's called vatiquinone. Come to that later. Again, very broad population, 6 months. And the paper manages to not mention the change from baseline in the placebo group, but you can actually infer it from other numbers. So it was about 2.0 points. And the natural history in this age changes by about 0.4 points. That's mostly or very much impacted by these old patients in the natural history study that show an improvement in the first year or so. What the paper shows, however, is a 24-month change in mFARS when they did an extension study. And it's seemingly the treated patients stayed at that -2 points, and the natural history supposedly declines by 5 points. So that analysis is not good. I'm not going to say it any worse, but I can't imagine a subgroup that declines by 5 points that would be comparable to this age. So I would think this population declines maximum 1.5 points in these 2 years. Whatever, if we look at 1 year, again, we end up with this round about 2 points. 6 months, not 1 year. Sorry about that. Now, the first study that actually used the mFARS was the interferon gamma study. A little bit younger population, 10 to 25, and a change from baseline in the placebo group by about 0.8 points. Natural history declines about 1.1. Again, we are roughly at 2 points. Okay, I think I know what you all think. This is really, you know, is this really worth something? And that's exactly what I thought when I saw these numbers and thought, what do I do now? I think it's a jungle. We really do not know how to compare these, and it's really difficult from the data we have to come up with good results. So before we can clean this up, of course, there are all these things that we all have in mind, and I wonder if I should repeat them to you. I only compared age-based subgroups, which is the most important determinator, but it's not enough because none of these trials are stratified by age, which I think is a mistake and should be done. And other properties like genetic and functional severity are very important. They often enroll very highly heterogeneous populations, and the group sizes don't align with that. They're just not able to cover the diversity in these groups. Then they were too short. The time points don't really overlap in natural history studies. The big studies usually have 1 year intervals, and we have different patients. So I wasn't happy with this. So I went to the real data we have in the FAICD. As you know, there is a database where sponsors can donate their placebo group data. And for these studies, that's what's been done. And I've directly looked at those results. I made a few restrictions because it's necessary to get a good comparison. And I can explain those in detail, but for this talk, I've restricted myself to ages 12 to 40, and I looked at this mFARS 85 just to be able to compare more studies than just a few that actually used the total mFARS. And what you see here is the 2 natural history studies. So FA COMS declines about 1.5 points, and FA child, which is a more severe population, declines a bit more. Makes completely sense. Now, the first study was Ionia. These were also children. And you can see that the effect is very strong after 3 months and goes away already after 6. And that's why many people believe if that study had went on for 1 year, we would probably have idebenone as the first drug in FA. Second study is the Steadfast study. These people were a little older, a little less severe, and very similar after 3 months, and then moved closer towards the natural history trajectory. The EPI study, again, people less severe, more placebo effect, and less return towards natural history trajectory. Now this looks, patients get more severe, they might show more less severe, I'm sorry, when they show less severe genetic severity, they show higher placebo effect and it takes them longer to return. I think that would make sense. The problem is if you look at Mykonos, which is clearly the least severe, you don't see anything here. So I'd like to not show this, and I have an explanation here, but it's not the case. So what we also found recently, or what's been shown recently, is that a lot of this placebo effect comes from the upper limb, or I should say appendicular part. You see here very similar effects in this subscore of the mFARS. And yeah, I'm just going to going to skip on to the next, because the interesting part is, of course, the upright stability, where we see very close trajectories in the natural history study. Then we look at Ionia, very close to it. We look at Steadfast, a little bit of a dip here, but then back to natural history already at 6 months. Epi, increase all the way. Mykonos, just the same. This is what we need to look at. There is very little placebo effect in the axial portion of the mFARS. And that is the lower limb part that is also appendicular and shows a similar pattern to there. So I'm going to skip those and summarize. So I think the placebo effect is highly variable also in these RCTs. It's difficult. We're not there yet where we can say we can do a reliable model based on this data and then tease the effects apart. But I will say that the axial function is clearly less effective. I hope I convinced you of that. And then, of course, it seems age-dependent. If that wasn't that Mykonos part, it seems to become less over time. And the understanding that we have over the thumb, it takes about 1 year to reach the natural history decline again. Now, I just like this slide, so I need to put it again. We have the elephant in the room in FA that we always need to mention. In their trial, what you can see is the same thing. The mFARS score shows a placebo effect, an improvement after 3 months, and then the placebo group follows the natural history trajectory. In the upright stability part, you can argue there's maybe a dip here, but in essence, this line is flat, and the placebo group declines close to what we would expect from natural history. And a final slide, and then I stop talking. In MOVE-FA, the study that was just published, the main endpoint was missed, although the difference is quite big, I would say. While in the upright stability section, the endpoint was met. And I bet, I don't know more data than this, what is public, but I bet that this is in part due to the placebo effect that we've seen in the upper limbs. And here I close. Thank you.
Thank you very much, Christian, for this talk. There are already a lot of questions in the chat, and I'll start with one I also had. This stability of the upright FARS towards placebo effects. Do you think that is something that is generalizable also to other forms of ataxia?
I think it's enough examples to argue that it is generalizable, yes. These trials are very much independent. Independent tests of each other. So, and the MOXIE trial again confirmed it. I'm still curious what MOVE-FA really showed in the progression, but I think it very likely will confirm this.
These are all Friedreich ataxia. So
these are all Friedreich ataxia, yes, but what we see also in the natural history, and that is similar to what also Biohaven has said about their natural history analysis, that most of the, now let me think that I don't say anything wrong. Most of the decline in the natural history study comes from the axial part. This is also a reason why the FSARA is, well, some people prefer the FSARA. It only looks at the axial section. And the appendicular parts show higher variability, they show training effects, there's questionable relevance of them. And all this together, I think, makes a strong argument to look at the upright stability specifically. On the other hand, if you look closer at the READA trial, their overall effect was like 2.5 points, and the axial effect is only 0.9. So they made potentially more than a point of effect in the upper limbs. So a big portion of their effect comes from the upper limbs where there is no decline in the natural history. So I don't, that's the only argument I have why we should still look at upper limbs.
Hey, Christian, it's good. I just wanted to, I apologize for being late. I missed the email invite, but I just wanted to quickly comment. That's what we've seen looking at both CRC-SCA and Euro-SCA, that the axial items, whether it's pure just axial items extracted from the SARA or F-SARA, which is obviously limited to the axial items, do show a steeper trajectory than the appendicular items. There is some progression, though, in the appendicular items. To your point, I think that we're seeing, but the magnitude of change in natural history is far less than the axial items. And one of the things we did see is, and we've actually presented some of that now. The trajectory for the SCA3 at 1 year in the placebo arm of 206 was quite similar to the trajectory for the natural history at year 1 in terms of CFB, in terms of change from baseline for the again what would we call the F-SARA. So
I'll read one question from the chat, and then Sylvia, you are next. So one question from Matthis is, Christian, you mentioned that that age is the main discriminant of progression. Is it really age or is it severity? Because the two are, of course, very correlated. How can you tell it's really age?
Oh, that's a difficult, I'm going to talk for 10 minutes if I start with it. But this is clearly an FA feature. So this is not generalizable to the SCAs. I believe that also in the SCAs, younger patients are potentially more severe, but it's clearly not as strict as it is in FA. So that's not, I made this point so people would understand why I look at age-based subgroups and why that makes sense in FA. And we by now have a really good feeling how people in different age groups behave in FA. In the SCAs, I'm not able to get my head around it. It's really difficult. I tried these kind of analysis just to understand it better, but it works nowhere near as clear as it does in FA. So,
Sylvia, you're next.
Yeah, just a comment. In Friedreich's ataxia, of course, you have the legs and walking, standing, and upright position, as because this is really completely different from the SCAs clinically. So and you have the problem if you have lost or you are near to lose gait or stance. In Friedreich ataxia, it's very difficult to get it back, because this is this will not come back if it's gone. So if one defines the group, and in Friedreich ataxia, the other SCAs are mainly the repeat SCAs. Let's say there is just SCA2, which has early beginners, which are children, and the other SCAs are not existent in the kids, basically, not in a relevant group size for the repeat SCAs, so for the major proportion of SCAs. I think one should keep that in mind, and and the problem is to measure stance and gait, and improvement of that is very, very, very difficult. And this has also been shown in studies, for example, botulinum toxin for leg spasticity and things like that. It's really, really because force, stance, this is a mixture of different abilities that are impaired. So this is for gait. So I think one should not talk about ataxias and include Friedreich ataxia. Friedreich ataxia has to be discussed completely apart because of the age and because of the clinics. And for the cerebellar SCAs, they are not existent in kids or very young people, and so you will have adults. And the second thing is you have in all these scars, so the major three, 1, 2, and 3, a mixture between cerebellar and extrapyramidal or pyramidal disorder. It's not pure. So I think one has to look very carefully, and even if we don't have very many patients, we have to stratify and we have to stay with the disease and not with this is all ataxia.
No, totally agree. And a comment on this loss of ambulation. What I have learned as a statistician, you all know that, is that the loss of ambulation is much more aggressive in Friedreich. It's more a weakness, a muscle weakness, loss of ambulation, and that's why the trajectories, if you compare ambulatory and non-ambulatory patients, are so different in FA. Using the SARA or the mFARS, it's the same. However, if you look at the scars, and I can't pick out a specific one, but it seems to me that in the big natural history studies, these two trajectories are different, but not by far not as different as they are in FA, because the upper limb items are lost earlier and the decline goes much more, is concomitantly the right word. So they all decline in the same fashion. While in FA, first everything is about walking, and only when they're non-ambulatory, upper limbs start to decline. So that is an argument that this might be slightly different in the scars, but I still believe it will be similar.
I don't want to jump ahead of everybody if there are further comments, but again, looking at the natural history, we did see, sort of the more ambulatory subgroup, baseline gait 1 or 2, had a steeper trajectory than overall. And again, this is on the CFB for axial items for the FSARA. So yeah, it will be of interest, Christian, to run some comparisons between both Friedreich's and also the ataxia data, natural history sets. Yeah.
Just another question from the chat on the placebo effect in the upper limbs. Do you think this is more a property of the clinician-graded scale, which could be remedied if we find other means to quantify that, for example, with a digital motor outcome? Or is that truly a property of upper limb function and the disease?
Does that go to me?
Goes to you or anybody else who feels
I think it's a property of the test, because these are really not natural things to do. And the mFARS upper limb part is not different from the SARA upper limb or appendicular sections. These are not things that people do in everyday life. But if you look at stance and gait, that's what they do every day. They try to keep their balance every day. That's what they struggle with. They will train it. And there is no, just because of that, there might be no training effect in these items or placebo effect in these items, because they struggle so much to do that every day. With these upper limb things, I think that's completely different. And that would explain learning effects or placebo effects, because they just try harder in a clinical trial than they do in the natural history.
And that would argue in favor of finding tests that more reflect really everyday function of upper limbs.
Absolutely, yeah. And they exist. They're just not as simple as a scale maybe is to handle. Yeah.
Are there other questions or comments?
I can comment to Michelle, someone asked about the baselines. That was one of my worries, that actually the effect you see in MOXIE is, or I should start differently. If you think about upper limb function decline, then you will of course restrict your population to those who have still room to decline. Especially in Friedreich, in every population or every study, you will find people that are close to loss of ambulation, which means they're close to maxing out all these items. But I was able to look at the baseline of these, and that was not the problem here. So I trust this effect. There might be one or two who are close to the ceiling, or if I'm not going to put a number in, a few, but the number is similar in both groups. So this is something I really trust, this effect. Now what else did I see?
Thank you. And you made the comment that they had room to decline. So it's not that. So thank you.
Yeah.
All right. They don't
Their baselines were in 22, 24 or so, and the maximum is 36. So that was the center of the distribution.
Are there any more questions? Now is the last chance for today.
Yeah, hey, Christian, I think, we've talked about this before. I think, if the sample allows, and I think in FOCOMs it may, the opportunity to do sort of multivariate predictive model, looking at, some kind of a generalized linear model where you could look at multiple variables at the same time and see how each age group, gender, repeat length, status, how predictive they are trajectory, adjusting, holding all the others constant, essentially, which is the beauty of GLM. I think there's the power to do that in, not sure if it's there, and I think it's probably there in combined natural history for SCA, but certainly for FOCOMs, you may have it.
All right, then I'd like to thank both speakers again for excellent and insightful talks, and also all the people for their time and discussions. We have one more studio series in this, or one more talk in this session, and that is on June 16th from 4 to 5 o'clock. So Matthis and us will be the speaker modeling placebo response in ataxia trials. Then I wish you all a nice afternoon and a good weekend.